Good morning. Myself, Dr. Mrs. Damini Motwani, Assistant Professor, Department of Biochemistry, Dada Ramchand Bakrus, Sindhu Mahavidyalaya, Pachpavli, Nagpur. Today I am going to talk on the topic gram staining. In the late 1800s, Christian Graham observed that some genera of bacteria retain a diiodine complex when rinsed with alcohol, while the other genera were easily decolorized with alcohol and could then be visualized by the contrasting counterstain. This staining procedure defines two bacterial groups. Those which retain the primary stain were known as gram-positive bacteria, and those which were easily decolorized were known as gram-negative bacteria. The difference in the dye retention is dependent on such physical properties as thickness, density, porosity, integrity of the bacterial cell wall, as well as to some extent chemical composition. Gram-positive bacteria have thick, dense, relatively non-porous wall, while gram-negative bacteria have thin walls surrounded by a lipid-rich membrane. Some non-bacterial organisms with thick cell wall also stain gram-positive. Gram-positive bacteria which have lost wall integrity through aging or physical and chemical damage may stain gram-negative. Now the staining procedure has various steps. The first step is the preparation of smear. Suspend some of the material to be stained in a drop of water on a microscopic slide. Spread the drop to the size of a nickel. Allow to air dry. Heat fix by gentle warming above the flame and other heat sources. The second step is to stain the slide with a primary stain crystal violet and then to wait for about 30 seconds or 1 minute. The third step is to add the fixative, which is also known as mordant, and here it is iodine. Flood the smear with iodine and wait for about 30 minutes, 30 seconds to 1 minute. The next step is to rinse. Rinse with water to remove excess alcohol. The next step is the decolorization. Drip 95% alcohol across the slide about 5 seconds. The effluent should appear pale or clear. The next step is again to rinse with water. Rinse with water to remove excess alcohol. The next step is to add the counter stain. In case of gram staining, the counter stain is safranin and then to wait for 30 seconds. The last step is to rinse, dry and observe. Rinse with water to remove excess stain, blot dry and observe under the microscope. After finishing the staining process, the slide is observed under the microscope. The microscopic view of gram staining is shown in the figure. Gram-positive rod and cocci will appear as violet because they take the primary stain, and gram-negative rod and cocci will appear as pink because they take the counter stain, that is safranin. Thank you.